Well, good morning, everybody. Thank you for being here at 9. First of all, I want to thank the organizing committee for their kind invitation to come here. It's always a pleasure to meet a lot of friends. And my role this morning is to actually introduce you this new field, or relatively new field of the CRISPR-Cas system. And I'm going to try to do that using one of our bug of interest, which is Streptococcus thermophilus. And also, although I love phages quite a bit, actually here we're trying to sort of get rid of them. So it's going to be a bit different, right? So this is the outline of my talk. So I'm going to give you a brief introduction on the CRISPR-Cas system. And this system really works on three main stages, and I'm going to hopefully slowly, present them to you. And then obviously I'm going to conclude with some perspective as well. All right, so I don't have to go through this slide quite very long for you guys. You're fully aware of virulent phages. Actually, in my talk, I'll be mainly talking about virulent phages. So they will inject their DNA into the cells, and they take control of the cells and start producing their proteins, and then will assemble and then lyse the cells. And this will be done pretty fast. In Strep thermophilus, this can be done in about 30 minutes at their optimal temperature of the cell growth, and about 100 new phages will be released per infected cells. What's not that well known is that bacteria do have a number of phage resistance mechanisms. They're classified based on how and where they're acting into this phagelytic cycle. Although today I'm going to talk about the CRISPR-Cas system, which is a mechanism that actually cleaves phage DNA. All right, a little bit of history on the CRISPR-Cas system. First of all, this structure, or this repeats and spacer units, were discovered in 1987. While a group in Japan was studying the coli genomes, and they, as you can see here, maybe, they found when they were sequencing one of the genes in 1987, obviously it was more difficult at the time than it is today, they did find these repeats, and they had these variable regions, and they didn't know at the time what they were doing. And now these variable regions are called spacers. And it's only in 2002 that actually an acronym was proposed for this system, which is cluster regular interspace short palindromic repeats. That was proposed by a Dutch group, and actually that's the one that's been used by the field now. There's a number of other acronyms that have been proposed over the years, but this is the one that stuck, and it's the one in use. So how it works, you've got this CRISPR, what we call CRISPR locus or CRISPR array, where you have these repeats, which are these black diamonds here that are represented by black diamonds. And these colored rectangles represents the spacer, which are variable regions. And upstream or downstream from this array, you've got genes, what we call Cas genes for CRISPR-associated genes. And those repeats will vary in size depending on the bacteria and on the systems, and the spacers as well will vary in size as well. And now we know that these variable regions are actually matching phage genome sequences or plasmid genes, and some of them, as we'll hear this morning, can match also some host genes. Most of the CRISPR-Cas system are chromosomally encoded, although some of them have also been found on plasmids. In Strep thermophilus, we did a study a long time ago, in collaboration with Danisco, where we look at the spacers in the number of Strep thermophilus strains, and we realized that a lot of these spacers, those variable regions, were matching phage genomes, and some of them again plasmid and so forth. So now we're able to actually characterize Strep thermophilus strains based on their CRISPR array and the content of these spacers sequences. The Cas genes also that are, like I said, upstream or downstream this array, they're highly variable, and I'm going to show you a slide in a few seconds, in the gene content and organization. There's although two genes that are very important, which is Cas1, which is universal, which is found in all the CRISPR-Cas system. I mean, if you find an array, you're going to look for a CRISPR array, you're going to look next to this array for this particular gene, a Cas1 gene. And in most of the system, you'll also find a Cas2 gene as well. And there's a number of studies that have been done on these Cas genes, and they have been classified in families, and a number of these proteins have been characterized, have been crystallized, and all the biochemistry is going on right now on these proteins, and some of them are involving DNA, RNA binding, and helicase, endonuclease activities, and so forth. So this is how we have classified at this time the Cas genes. And they have been classified pretty much in the same way I classified a restriction enzyme that you all know, which is in type 1, type 2, of type 3 system. And you'll find in these different rectangles here in colored, those are signature genes for these particular types. So it's essentially you've got your CRISPR array, you look at the genes are flanking it, and then you're trying to find these signature genes that will allow you to classify your CRISPR-Cas system into one of these family. Today I'm going to focus on the type 2A, which is the one that is very active in Strep thermophilus. But because of this diversity of Cas protein that are next to these CRISPR arrays, there was a lot of question about whether they were working the same way, right? Because there's a lot of different genes, different proteins that are involved, and are they really working all the same? And that's a lot of people are actually studying this right now. Thanks to a group of Christine Pourcel, which is in the audience, there's a lot of bioinformatic tools now to know if there's a CRISPR array or CRISPR-Cas system in your bug of interest. So if you're working with one particular bacteria or archaea, you want to know, is there a CRISPR in this bacteria? You just go to Christine's website and just go through, and the genomes have been looked, and it will tell you if there's a CRISPR or if there's many CRISPR arrays in your bacterial genomes. And what those bioinformatic tools told us, though, is that about 45% of all the bacterial genomes that have been sequenced to date carry a CRISPR-Cas system, and over 90% of the archaea as well carry this type of mechanism. So it's fairly common in archaea in bacteria. So this system works in 3 main stages. I'm going to go through them. First, it's called a spacer acquisition system, or if you want, we can call it also adaptation or immunization system. I'm not going to talk too much about biogenesis of small RNA, just a couple of slides, because Emmanuel after me will go in through the detail of this. And of course, I'm going to talk about the inference as well. All right, so the bugs we use for this, most of our studies on this system is Streptococcus TGG7710. It is an industrial strain in the sense that this bacteria is used to make yogurt, to make a variety of cheeses. So when you're making cheeses or yogurt, you're going to heat-treated your milk. Your raw milk is going to be heat-treated. Again, you're going to add bacteria, and Streptococcus is one of them that you can add to start your process of milk fermentation. And of course, in this particular industry, phage are a problem, so the idea is to have bacteria that are resistant to phage, so you can have your process that will go properly and make your nice products that hopefully you're eating. And this particular strain, the TGG7710, is actually phage sensitive. It carries no plasmid whatsoever. We do have the draft genomes, and when we studied the genome of this particular strain, we found 4 CRISPR loci. Oops. And these are the 4 CRISPR loci. You've got CRISPR1, which is a type 2A, CRISPR2 type 3A, CRISPR3 type 2A, and CRISPR4 type 1E. In this particular strain, the CRISPR1 and the CRISPR3, both of them are type 2A, are the one that are really active, and that's the main focus of my talk. So CRISPR1 carries 32 spacers, it carries 33 repeats, whereas CRISPR3 has 12 spacers and 13 repeats. And you've got the Cas genes that are next to it, of course. And right away, by looking at the number of spacers, one has 32, one has 12, you can already tell that this CRISPR array looks like to be more active than the other because it's got more spacers. So we also use phage genomes. We have a number of phages that are infecting Strep thermophilus. We have done a lot of phage genomics on this. So the 2 phages that we are studying are 2972 and 858. And in Strep thermophilus, we don't have the diversity that we are finding in Mycobacteria phage, for example. If you look at Strep thermophilus phage under an electron microscope, this is what you will find, a Syphoviridae type of phages. If you sequence their genomes, you can classify them in 2 or 3 clusters at the maximum. So there's a lot of, so they look alike quite a bit. But there's obviously some differences, particularly in the area where the genes that are coding, there are early expressed genes, whereas they're more homogeny or similarities, should I say, in the protein, structural protein. So the experiment's pretty simple already when we started. We used the phage-sensitive strain, we had the phage in a broth, and then we put that on a plate. And then you incubate at 42 in case of Strep thermophilus, 42 Celsius, and then you wait. And then you get colonies that will start to appear. And in Thermophilus, actually, most of these bacteriophage and sensitive mutants of these colonies that will appear are actually their resistance to phage, but also that resistance is derived through the CRISPR-Cas system. So in thermophilus, it's very difficult actually to get a receptor mutation or any other type of mechanism. It's really the CRISPR-Cas system that's very active. It's the most active actually. So for example, if you do that in E. coli, most of your BIMs or bacteriophage-sensitive mutant will be receptor mutants, right? But in thermophilus, they're CRISPR BIMs. So that's a big advantage if you use this system. So if you take those colonies, you streak them, purify them, and then retest for phage resistance. So you got the wild type here. The phage was serially diluted, and you could easily that this bacteria is sensitive to the 2 phages, and you've got this this colony. If you grow it in broth and then do it on a plate again, then it's resistant to one phage and sensitive to the other, and if you got this other colony, it's resistant to both phages. So the question we were asking what happened, and because we had the draft genomes, we know where their array were, and we had PCR. We could design primers that were targeting some of these spacers here, and you can do PCR. So you take the colony, do PCR, and we saw an increase in the PCR product. The size of the PCR product actually was 66 nucleotides. So we obviously sequenced that, and it was 30 base pair for the spacer and 36 base pair for the repeat. So there was an acquisition of 66 nucleotide, or base pairs, sorry, in the CRISPR array. So of course this was done in a test tube. So it was pretty easy to figure out where that DNA came from, that increase in size. So when we sequence, we look at the spacer content, and we did some blast searches. And this particular strain, which was sensitive to one phage and resistant to the other, had acquired a piece of DNA from the phage genome. And that piece of DNA was not present in the other phage. And on the other hand, you had this BIM here, which was resistant to both phages, had acquired a piece of DNA from the phage that we used for the phage challenge, and that same piece of DNA was perfect match in the other phage genome as well, explaining why this bacteria was resistant to both bacteriophages, whereas here it was only resistant to one. So we had to come up with a differentiation come up with a name, or how we would call the phage DNA. So when I'm relating or talking about the phage DNA, we call it the phage protospacer, and when we're talking about the spacer, the variable region in the CRISPR array, we call it a spacer. So we have done this quite a few times, that phage challenge assay. We have purified these colonies. Actually, there's if you take that colony here, for example, and you streak it and look at the CRISPR array, there's actually a war going on. There's a lot of phage in the plate, and there's a lot of CRISPR, a lot of spacer that have been acquired and so forth. So you really have to purify your colony to really make sure that you have a stable CRISPR array, and then you can study that system. So one day maybe we'll be able to go into that colony and see what's going on, but it's really a big war between the phage and the bacteria. So what we did recently, we've done this 320 times and tried to see where that space, what was going on with the CRISPR-Cas system, and in 280 of them, they were a new spacer that was acquired in the CRISPR-1, and 30 of them was acquired in CRISPR-3. And we were lucky to get a couple of BIMs that there was an acquisition in the CRISPR-1 and the CRISPR-3. So we had a colony that had acquired 2 spacers, one on both systems. In some cases, we also had the deletions of the arrays. So some of the bacteria were actually acquiring a spacer, but they were losing some of them, especially the ones in the middle of the array. And then most of the BIMs were resistant to phage to the acquisition of 1 spacers, whereas others were resistant because they had acquired multiple spacers. We can even have one that had acquired, in one challenge event, had acquired 4 different CRISPR new spacers. So what's really interesting here is that we have done this. We have picked 320 colonies. We have characterized that, we purified them, and then if we look at the spacer content, and it turned out that if you look at the phage genome, we have spacers that was acquiring that were targeting. We had 6 spacers that were targeting gene 1. So what I mean by that is that a spacer that came from gene 1 was acquired in the CRISPR array, and so forth. So pretty much we have BIMs that are sort of covering almost all the genes in the phage genome. So we're going to be studying this now to see if we can use this to mutate the bacteriophage. Now, as I'm going to show you in a few minutes. I'm a Mac user, can you tell? When we have looked at the so we had this CRISPR array, that all these BIMs that have acquired spacers into their array. So we were trying to find some sort of something. There was something that was conserved in all the spacers that have been acquired by the array, but we could not find any motif, any domain, except if you look at the phage. So all these sequences here that are spacers that have been acquired by the host. But if you look at the phage genome, which is the exact same match, next to it, there's always this motif here called the PAM motif, or protospacer-associated motif. And this motif seems to be really important for the acquisition stage and also for the interference. In the phage genome of 2972, there's 231 PAMs that are specific for the CRISPR-1 system. What I mean by that is that all these spacers were acquired in the CRISPR-1 array. And if you look next to it, this motif is found in 231 region of the phage genome. And what it actually means is that there's 231 possibilities of acquiring a spacers in the CRISPR array, CRISPR-1, from that phage. So there's 231 possibilities. And I think in the lab we have 170 of them. So we can pick them up pretty easily. So again, in the bacteria, you've got the spacer. In the phage, you've got the protospacer, and you've got this motif that is next to it, all right? In the bacteria, next to it is the repeat. Okay, so there's a difference right there. You probably did not see it at the back, but if you look very closely, if you used to work with phage, when you see little dots here, you know it's a plaque, and you know it's a phage mutant. So we pick that plaque here in the Sparkler system and then amplify the phage, sequence its genome, and there was only one mutation out of 35,000 base pair, one mutation. And where do you think it was? That's the question. In the protospacer, of course. So there was a mutation. So again, the host spacer, the bacteriophage, protospacer, and the motif. And you've got 3 phage mutants here that are shown. And there was a mutation either in the protospacer or in that motif. And that mutation alone was enough for the phage to bypass the CRISPR system, meaning that if the bacteria acquire a new spacer, if you have a mutation in a CRISPR array, if you have a mutation in a protospacer, then the phage can bypass the system. And actually, when you do this, you can actually get deletions as well. So that if the gene is non-essential, you can have deletion of the phage gene. So it means that phage mutant can bypass the CRISPR-Cas system with a point mutation within the protospacer or the PAM. So what we did is we took our old BIM, take the phage mutant, and do the assay again. And sure enough, we got, again, phage-resistant mutant. And in this particular case, you got the Cas genes, the array here, repeats, spacer repeats. You've got the first BIM that we acquired, that acquired a spacer called S4. And in the second iteration or second experiment we did, it had acquired a second spacer. And it's very interesting, if you take the wild type phage and test this wild type phage against that strain is really, really resistant to that phage. So if you acquire one spacer, you're resistant to phage, but if you acquire 2, you're even more resistant, and so forth. So what we come up with is that what we thought was that the spacer represents some sort of molecular bookkeeping, a memory of past phage infection. And this memory allows the cell to defend itself against phage infection. So how was the question? In the interest of time, I'm not going to talk about this, but just want to tell you that we are also able to, if you introduce a plasmid into that strain, grow that strain without selective pressure, the CRISPR array will pick up DNA from your plasmid, all right, and become resistant to that plasmid in the sense that that strain will lose the plasmid, and if you try to reintroduce the plasmid, you won't be able to do that because that CRISPR array has acquired a spacer targeting that plasmid. All right, so bacteria with an active CRISPR-Cas system can acquire a new spacer for foreign DNA, such as phage genomes and plasmids, and by an unknown mechanism. So a lot of people are trying to understand what's going on, how this is happening. It's a very active field. Biogenesis of small RNAs. So I'm going to summarize this in 2 slides, and then Manuel will have a bunch of slides. So you got, again, the genes, the array. There's a couple of promoters for the genes. And also there's a promoter in the leader regions here. So you've got this transcript of what we call a long CRISPR RNA. it's not an mRNA because there's no genes in there. It's a long RNA, and this will be processed by Cas protein, some of the Cas protein in very small CRISPR RNAs, and they will be processed, and at the end you'll have a mature small RNA that will guide and will be in the intracellular of bacteria and will sort of be looking for foreign DNA when it's going to come in. And there's been a lot of studies in that area, actually, a lot of really nice papers that have been, and what is happening is when we think that there's about at least three types of mechanism by which the small RNA are produced. Either it's a type 1 or type 2 and type 3, and like I said, Emmanuel will talk about a type 2 in a few minutes. All right, so I'm going to jump right away to the interference. So there was a couple of very nice papers that was Cell front page by the group of Michael Turns that were showing that in archaea, some of the type 3 CRISPR-Cas system of Pyrococcus was targeting RNA. This was a very nice paper, and it was done in vitro. And Luciano Marafini and Eric Sonheimer also showed in Science that it was probably targeting DNA, plasmid DNA in Staphylococcus aureus. So in two type 3 system, one it was targeting RNA, one it was targeting DNA, and really nice work. So we're asking ourselves what's happening with the type 2A system that we are studying. Is it DNA? Is it RNA? So we put the hypothesis that phage DNA was actually the target of these small RNAs that have been produced. And if it's the DNA and it's probably going to cleave it, where it's going to cleave it and how it's going to cleave it. So again, we use our Streptococcus strains. DGG7710, we use our BIMs that we had generated in previous experiments that were resistant to phage. So you've got the wild type with the 32 spacer, you've got one BIM that's got S4, second BIM S7, and you've got this one that has acquired 2 spacers. So these BIMs have the spacers were acquired from different part of the phage genomes. Some of them was targeting probably a genes that was coding for a structural protein, and whereas you had genes targeting genes that are expressed early when we didn't know the function. So experiments, pretty simple again, in our hands at least. We have the infection, we grow the cells, then we concentrate them, we add the phage and MOI of around 5, and then we take a sample, and then we follow over time, and we take sample at 2, 15, 30, and 45 minutes, isolate the total DNA, restriction digestion of the total DNA, migrate on agarose gel into a southern blot using probes, specific probes. So this is a fairly complicated slide, but I'll try to go slowly, and hopefully I'll make sense out of it. So this is again the phage genome. You've got S pro spacers here, S7, S4, S32. So if you digest that phage DNA, enzyme HPA2 here, for example, you're going to add a 7.3 kb fragment. And if it's cleave around the protospacer region at 7, you should get a 3.2 kb fragment and a 4.1 kb fragment, and so forth. And the same for the other region. So if you use the wild type strain, you infect with the phage, of course, you'll see an increase in the phage DNA replication. And so this is the probe here targeting this region. The gray boxes are schematic representation of the probe. So here, this is the wild type. At 2 minutes, you see that this fragment starting to appear and so forth over time. And the 3 different BIMs the different probes that we use show the same pattern. Jeez. All right, so, and sure enough, in BIM 7, as you can see here, you saw the appearance of a 3.2 kb fragment. It was appearing, showing clearly that the phage DNA was being cleaved by the CRISPR array. And you see here also we had the 4.1 kb fragment that was appearing. Very surprising to us was the presented on-cleave molecule into those assays. Although there's a lot of DNA on that southern blot here. There's about 10 times, at least 10 times more DNA here than here. So a lot of phage DNA is actually cleaved, been degraded, but some of them was still there. And what's very interesting here is because in this BIM, you remember that I've acquired 2 spacers that were sort of next to each other. Here. If it was cleaved in the protospacer region, you would get a small fragment of about 1.4 kb, and sure enough, we're able to see this small piece of DNA. So a strain that has acquired 2 spacers could cleave phage DNA twice. So we had the DNA. Next step was to see where it was cleaved. And it would cleave right within the prospacer at 3 nucleotides next to the PAM in the phage genome. So the CRISPR-1, the type 2A, was cleaving 3, was cleaving in this region. So we've done this with multiple BIMs. Every time it was cleaving in the same area. We also did that with a strain that had acquired a spacer targeting a plasmid. And again, it was cleaved at the same region. We now recently done the same type of experiment using BIM that had acquired spacer in the CRISPR-3 system, which is another type 2A system. And again, the cleavage was at the same space, same place in the phage genome, which is 2 nucleotides next to the PAM. So we're really, really convinced that the phage DNA is cleaved in the prospacer region, in this specific region. So conclusion perspective. So how it works, how we think it works, or how we know so far. Phage DNA goes in. Somehow Cas proteins are involved. At least we know some Cas proteins are involved. Cas, CSN2, Cas1, Cas2, and CSN2 are involved, most likely. We'll cleave this phage DNA. It will be integrated. It's always polarized. The integration is always at the 5' end of the CRISPR array. And there will be a repeat that will be duplicated, and with some really nice work done in E. coli that I've shown that it's the first repeat that's actually been duplicated here. All right, we don't know how this is happening, but this is happening. Then after that, depending on the type of Cas genes that are next to this CRISPR array, the long RNA, CRISPR RNA will be cleaved and to produce small CRISPR RNA. And this CRISPR RNA will be able, by base pairing, go and match the double-stranded phage DNA. There's an R loop that will be produced, and then this will be cleaved, and Manuela will show you how this is done. Not Emanuela, Emmanuel. So there's already commercial application for the CRISPR-Cas system, and Denisco DuPont, who's been a long-time collaborator with us, have already introduced BIMs to make cheeses using the CRISPR-Cas system. And this is natural phage resistance, because as I showed you, you can take a phage, say the bacterium Streptococcus, select your BIMs, and if you do it multiple times with different phages, you're going to acquire multiple spacer, and you're going to have a very phage-resistant strain. There's a number of things that we still need to understand. What are the exact role of Cas protein, how spacers are acquired, as I've told you. Do CRISPR-Cas system play other roles in the cells? We don't know. There's some evidence that they might. Are CRISPR-Cas system compatible with other phage resistance mechanism? I've told you there's a bunch of those in bacteria, but are they compatible with them, and so forth. And this is very interesting because this field has exploded since 2007. So this is a number of publications since 2007, around 10. And as you can see, it's going pretty fast. And obviously, another citation also. So it's a career opportunity for young scientists in this room. They were looking for a field to go in. That's not bad. And obviously, I want to acknowledge a very nice group of talented people. First of all, Hélène Deveau, which started this work a long time ago with us. Josiane Garneau, Manuela Villon, Marie-Ève Dupuis, and Alfonso Maganan. Alfonso is with us. Also funding for NSERC, CRHR, and Canadian Research Chair. And of course, Denisco DuPont, which we have been working with DuPont for Denisco for over 12 years. And I want to thank Christophe, which is in the audience. Of course, geez. And I'm going to finish telling you that we have a tenure-track faculty position in our department that is open in microbial virology. I wanted to make it virus of microbes, but that didn't go through. But anyway, so there's a deadline for if you want to submit your CV. It's September 14th. On that, I'll be happy to answer any questions. Thank you very much.
So, thank you, Sylvain. I think there's room or time for one question.
Get it. Sylvain, from the point of view of the bacteria, CRISPR system is not so efficient, right, to resist to a new phage. What is the proportion of bacteria that become resistant?
Yeah. I skipped through. That's a good question, and Marie-Agnès. Actually, if you take about 1 million cells, about 1 bacteria will acquire a spacer. In Streptococcus, that's about 1 of 1 million, 1 out of 10 million. So it's not very efficient, and that's why probably we don't know how it works yet, the acquisition part, because it's only one cell out of a million, whereas the interference, all the cells are resistant, so you can take your pull of the cells and then you see what's going on. Yeah, so it's very infrequent event for sure, yeah.
Okay, maybe one small question.
Eric?
So any idea why Cas1 was so much more favored in the resistance than Cas3, so much more frequent?
No? We don't know. I don't know, sorry.
Okay, thank you, Sylvain, for this nice talk.